Okay, I think we're going to get started then. Okay, it's a real pleasure today to introduce Bernardo Sabatini as our speaker. Bernardo is the Moorhead Professor of Neurobiology at the Harvard Medical School. It's actually a little difficult to describe Bernardo's research program because he has made significant contributions in more than one thematic area. He's done significant work on synaptic plasticity and control of neurotransmission, also some significant work on developing new methods. But I know him best for his work on both the structure, organization, and function of basal ganglia, for which he's made some very substantial contributions in recent years and has a very active research program. So, Bernardo, thanks very much for taking the time to speak with us.
Thanks, Roger. My pleasure to be here, and thanks for everybody for attending. Let me just bring up my slides. Okay, hopefully everybody can see that.
Yeah.
Good.
Well, let me just say one thing. For those of you with questions, I think for these Zoom talks, it works a little bit better if we wait till the end or put them in the chat so we can get back to them later, okay?
Sounds good, okay. All right, so like many neurobiology laboratories, we're interested in addressing the question which is posed here, which is how we choose what to do next. And we do that in a variety of contexts. We study always mice. On the left, you can see a mouse exploring a featureless bucket. This is work that's led by Bob Data, who's my neighbor at Harvard Medical School, and his lab, in collaboration with us, examines the basal ganglia contributions to the animal, choosing among the various different motor actions they can do in the absence of any kind of sensory stimulus. Most of what we do in the lab, however, concerns more the kinds of behavior shown on the right, in which we have mice that are performing some kind of task in order to obtain a food or a water reward. For example, in the middle here, a food reaching task, and on the right, a lick right, lick left task for water. Down below, you can see a mouse, in this case, a male mouse, that's exploring an arena and deciding when to release urine, so micturition. This is not a basal ganglia dependent behavior, we think, but also a form of action selection that we've been studying in the lab. And so we tend to ask questions like the ones that are shown here, which is, how does activity in the basal ganglia help us choose what to do next, evaluate if we made the right choice, and then update our plan to do better in the future? And now, we don't need to go through details of basal ganglia circuitry here. I'm sure many of you are familiar with it, and it's not incredibly relevant for today's talk, except to say that, of course, the basal ganglia consists of reciprocal circuitry with cortex and thalamus that forms these closed loops, and activity in these loops is thought to be essential for selecting an action, for triggering an action, and then later for modifying plasticity in the circuitry to change action selection in the future. And the kinds of questions that I talked about before can be overlaid into different parts, of the basal ganglia. Today, we're really going to focus on this question, which is whether doing something is predicted to be good or bad, right? And we're going to focus on circuitry within the mouse that we think helps evaluate that, and that in turn allows context and sensory information to regulate dopaminergic transmission in expectation of something good or bad happening. Now, we're a mouse lab, so I'm going to tend to use mouse nomenclature. And so, for example, we're going to talk mostly about the endopeduncular nucleus today, which corresponds to the globus pallidus internus in primates, and connections between it and the lateral habenula, which shows some very interesting features that are going to be the topic of today's conversation that we think are essential to evaluating this question of whether doing something will be good or bad. So the work I'm going to talk about was started long ago by Mike Wallace, who was a postdoc in the lab. Now he's leading his own lab at BU, and then carried on by Sola Kim, a former PhD student who did most of the electrophysiological recordings that I'll talk about, and is now being carried on by Shun Lee, a PhD student in the lab. But I'm going to warn you that what I've done here is taken sort of a mature talk on the front end, which is the work from Sola, and then linked it to the follow-up work that Shun is doing, which is very, very preliminary. But I thought it'd be fun to show you some stuff that's really, quite raw, but will show you the way we're thinking about a certain problem and the kinds of data we're trying to get. So Mike Wallace, when he was in the lab, did some single-cell sequencing of the endopeduncular nucleus, the equivalent of the GPI, which you can see here in a sagittal section of the mouse, sits in a myelinated bundle that links, of course, the striatum back to the substantia nigra. And so the endopeduncular nucleus in the mouse can be divided roughly into two poles. There's a rostral pole and a caudal pole that are differentiated by the expression of parvalbumin and somatostatin. So the parvalbumin expressing side, shown here in red, which is on the caudal side, consists of what one classically thinks of as a basal ganglia output nucleus. So these are parvalbumin positive long-range inhibitory cells that inhibit downstream structures and show a lot of spontaneous activity. However, the rostral side consists of these somatostatin expressing neurons, which are quite interesting and will be the focus of what we're going to talk about today. I'm not going to show you any work, but we have done a little bit of in situ hybridization in primate tissue and confirmed that these same kinds of cells do exist in primate GPI. Okay, so what interested us about these cells is that they form the link between the basal ganglia and the lateral habenula, and they have some really curious features. Now, since this is a Udall center, all of you, of course, are quite familiar with the function of the basal ganglia in motor control and how perturbations of basal ganglia circuitry contribute to Parkinson's and Huntington's disease. So this is classic sort of motor-centric view of the basal ganglia. On the other hand, the lateral habenula is really implicated in mood and in depression. And so this might be an interesting link in which one might think that the sort of motivation of doing a motor action and the expectation of motor action might be tied to the motor action itself. Now, the other reason that we are interested in this area is, of course, the lateral habenula has long-range excitatory neurons, but whose net effect is to inhibit dopamine neurons. And so as dopamine neurons learn to signal that something good has happened or that something good is expected to happen and modify the circuit in such a way that repeats that expectation, instead, the lateral habenula, by inhibiting that, learns that something bad might happen and inhibits that happening in the future. The other thing that interested us in the circuit is that these somatostatin neurons turn out to be neurons that Robert Malino's lab discovered a while ago, which are neurons that co-release GABA and glutamate. So here we have a very funny synapse in which an individual cell releases two opposing neurotransmitters, one that excites the postsynaptic cell and one that inhibits the postsynaptic cell. Now, of course, GABA and glutamate are well understood neurotransmitters in the mammalian brain, glutamate shown here on the right. GABA is made by the decarboxylation of glutamate by glutamate decarboxylases GAD1 and GAD2. And so these are similar molecules, but they're distinct, and we can recognize cells that are GABAergic by the expression of GAD1, GAD2, and we can also recognize both kinds of neurons based on the SLC or the transporter genes that are responsible for encoding the proteins that move these transporters from the neurotransmitters from the cytoplasm into the vesicle interior. Now, I'm not going to show you all the evidence that these somatostatin neurons that link the endopeduncular nucleus to lateral habenula truly do release GABA and glutamate. We've looked at this in many different ways, from single-cell transcriptional analysis shown here on the left, in which we can find clusters that have all the relevant genes for both GABA and glutamate release, to single-cell fluorescent in situ hybridization showing the existence of somatostatin cells that express, for example, GABA production enzymes and glutamate vesicular packaging enzymes. We've also done array tomography. This is a super resolution immunohistochemical approach that lets us identify individual synaptic boutons here of somatostatin neurons labeled by YFP and show that they in fact express vesicular GABA transporters and glutamate transporters. And lastly, we can look at electrophysiology. If we optogenetically activate these inputs, we get these kinds of biphasic currents shown here on the right, which is a rapid inward glutamatergic current followed by a slower outward GABAergic current. So these are bona fide GABA glutamate co-releasing cells. And so Sola, for her thesis, decided to ask a very straightforward question, but which is difficult to get at, which is whether at these synapses are GABA and glutamate packaged in the same synaptic vesicle, such that each fusion event releases both transmitters, or in separate synaptic vesicles, such that sometimes GABA might be released and sometimes glutamate might be released. So how can we tell these apart? Well, we can do some simple modeling and just think about the currents that we would see in each scenario. So for example, if there's an independent release scenario, if we activate one of these synapses at a time and look at the postsynaptic currents that are evoked, and if we hold the postsynaptic cell at an intermediate potential between the reversals for GABA and glutamate, we should be able to see sometimes a failure when no synaptic vesicles are released, sometimes a GABAergic vesicle released and therefore an outward current present, sometimes a glutamatergic vesicle released and therefore an inward current present, and sometimes both, these biphasic currents like I showed you before. And indeed, evidence for this kind of release mechanism of GABA glutamate co-releasing synapses in the habenula has been proposed and seen by Maricela Morales' lab in some very nice papers from Root. On the other hand, if each vesicle contains GABA and glutamate, then we would expect that sometimes no vesicle is released, but that every time there's a release event, we would see both an inward and an outward current, maybe a varying amplitude depending on the amount of neurotransmitter that's been packaged, but they should always be there. And in fact, Robert Malino's lab described the presence of spontaneous miniature synaptic events that had biphasic qualities, suggesting that each vesicle had both. Okay, so now to really tell these apart requires a little bit of nuance. And the reason for that is that if we look at the average synaptic current for the independent release model or for the co-packaging model, in both cases, the average should give us a biphasic current that has an inward current followed by an outward current. And so we really can't tell these different release events apart if we look at average synaptic currents, and this is averaging either over time by stimulating one set of synapses many times, or over synapses by stimulating many synapses at the same time. They're going to give us the same synaptic current. So therefore, what we need to do is really look at the variance, okay? And you can see if we just calculate what the variance of the currents on the left are from the ones on the right, we can see that they're going to have different shapes and different sizes. So this gives us a hint that if we look at trial-to-trial variability, we should be able to tell these synapses apart. Okay, so then the goal then is to develop an approach by which we're going to stimulate a single presynaptic terminal many, many times, record the evoked postsynaptic currents, and then analyze the statistical model and repeat this many times. So in order to do so, Sola and I built a microscope that allows us to address single synapses at a time and pair this with an optogenetic stimulation of endopeduncular synapses within the habenula. And so Sola expressed a variant of channel rhodopsin called O-chief in somatostatin neurons of the endopeduncular nucleus by injecting into the EPU for Cre-dependent virus and somatostatin Cre animal. You can see here the labeling site. And then we can cut an acute brain slice and identify in the habenula the terminals of these axons that are infiltrating the habenula, and then study the effect of stimulating them. And so we did this with a microscope that had built into it a digital mirror microarray, which is a very cool chip. This one's made by Texas Instruments that consists of tiny little mirrors, about 6 microns or so on a side, that can flip between 2 different positions. They can do so very, very quickly. And so by flipping these mirrors to an on or an off state, we can direct light to either go into the microscope and illuminate the sample, or to not go into the microscope. And so by focusing this array into the imaging plane, we can give arbitrary patterns of illumination in the tissue and find patterns in which we can stimulate single synapses at once. Okay. So the idea then is that we can perform a whole cell recording from a habenula neuron and then flash little blue spots of light under conditions in which we have TTX and 4AP, and you can ask me about this later if you like, but under conditions in which we can carefully calibrate the stimulus to activate a single synapse at once, and then ask, what do we see in the postsynaptic side? So here's such an example in which in blue here is the timing of the optogenetic stimulus delivered to one of these small squares under very, very low powers. And then in gray, you can see the appearance of these synaptic currents that consist in this case of an inward current followed by an outward current almost every time. Now you can see in the background that there are spontaneous events that are going on. For example, here on the left, a whole bunch of miniature excitatory EPSCs, and you can see here in the background some failure events. But I hope you appreciate that most times there's a release event here. There's an inward current followed by an outward current. Okay, so let's compare then the experimental data to the 2 models. Well, here are the individual currents that were stacked up together in the previous slide. And what you can see is that almost every release event, we have inward, outward, inward, outward, inward, outward. Sometimes a failure event here with spontaneous events, sometimes some unclear events like those here, but the clear events are all inward followed by outward. So then we can start doing some statistical analysis. So for example, in this gray window, we can just ask, what's the minimum current and what's the maximum current? So nothing except asking where's the minimum versus the maximum, and then plotting those versus each other as a function of trial. Now, I think you can see already on the left that something interesting is happening because the minimum almost always comes before the maximum, right? In red versus blue. And that already tells you that there's something interesting about statistics here. If it were just random currents, there's no reason why one should come before the other. And then if we plot the minimum current versus the maximum current, you can see that there's a nice correlation there, as one would expect for co-packaging of the vesicles. So we can then compare this to models and then in a formal way, do some bootstrapping, some non-parametric statistical tests, and ask, does the data conform to what we'd expect for independent or from co-release packaging? And so, for example, if we look at the correlation coefficient between the size of the inward current and the outward current, or rather the minimum and the maximum, we can see here that what we measured was a correlation coefficient that's very close to 1 from bootstrapping, and that's quite different than one would expect by chance, which is close to 0, of course. And if we look at the models, the idealized models, a perfect co-packaging situation with no noise and perfect recordings, we'd expect, of course, the correlation coefficient to be perfectly 1, whereas for an independent release model, we actually expect it to be negative because a large GABAergic current should occlude a glutamatergic current and vice versa, okay? But from this, we can see that, in fact, our data is consistent with co-packaging of both GABA and glutamate in the same synaptic vesicle. So that's one statistical test that we did for many, many different synapses. Another statistical test that we did is a very simple one, which is ask, what's the probability of seeing a GABAergic current and the probability of seeing a glutamatergic current versus the probability of seeing both at once? And of course, in statistics, events are defined as independent if the probability of seeing them together is the probability of seeing each one alone multiplied. Okay, so what we can do then is measure synaptic events that occur in this shaded region here. So this says there was an excitatory event, everything above the noise line. In this shaded region in blue, which is everything above the inhibitory noise line, or in this joint region. And you can see that how many, for this example synapses, how many times we get currents in each one of these different boxes. And again, doing the same kind of statistical analysis and bootstrapping, we can conclude that GABA and glutamate release are not independent. They're almost always seen together in a way that's not expected by chance. So we did several of these tests, and they all led to the conclusion that GABA and glutamate are packaged in the same vesicles. I'll just point out that most of the sources of noise that we could come up with would all tend to give evidence for independent release as opposed to co-packaging. And that makes sense because if we add any noise source, it's going to tend to drive and destroy statistical relationships, and it's going to give evidence for independent release, okay? And so what this means is that if we find evidence of co-packaging, we should believe it. But if we find evidence of independent release, we don't know what that means. Could be noise in our system, okay? So for every cell and every synapse that we analyzed, we defined a model axis in which we pooled several independent statistical analyses that summarized how good was the evidence between this ambiguous value of 0, in which we can't tell if it's independent release or if it's noise, versus co-packaging all the way here on the right. And so the example synapse that I've been showing you, which is really quite clear evidence of co-packaging, is the one that's circled here in the middle. So it's one that's really in the middle of our distribution. We had other synapses that have even much stronger evidence of co-packaging. So based on this, then we would say, oh, sorry, and one thing I forgot to mention is the better our recording quality, so the better the series resistance was, the lower the synaptic noise and so forth, the more likely we were to find evidence for co-packaging. Okay, so from that, we conclude that each vesicle has GABA and glutamate in it. Now, maybe some of you believe me, maybe some of you don't. We were, of course, a bit skeptical, to believe this because it's a bit of a weird thing. And so we thought, well, how can we look at this in a totally independent way? And we came up with a different mechanism, which is to simply lower the probability of release. And so as many of you know, if you lower calcium, for example, external calcium concentration, the probability of a vesicle fusing and being released will drop. There are many other presynaptic modulators that will also reduce the probability of release. So for example, at these synapses, serotonin is a very good presynaptic modulator that reduces the probability of release. So we reasoned if we reduce the probability of release, for example, from 80% to like 40% by a factor of 2, then the two models should show something very different. If the synapses co-package GABA versus glutamate, if we reduce the probability of release by half, then the probability of release of seeing these biphasic currents should also go down by a factor of half because there's only one event that's being regulated. On the other hand, if GABA and glutamate are released independently, then if we reduce probability of release by half, then the probability of seeing these biphasic events should go down by a factor of 4 because there's 2 independent release events that are coming together, and each one of those is being cut down by a factor of 2. So we did that experiment looking at single synapse release events before and after the application of serotonin. Here are the average currents before and after serotonin for a single synapse. You can see that serotonin scales both the inward and the outward current proportionally. These are small currents that you can see here, but it scales them both on average, of course, mixing failures and successes. But then we can look separately at each event and look at successes versus failures. And what I hope you can appreciate here is that the size of this inward current and outward current is unaffected by serotonin. It's just the probability of seeing the current. And that when we do see the current, we still see it as biphasic. And so without showing you all the mathematical analysis, this again confirmed that GABA and glutamate were in the same synaptic vesicle. We think the potential for serotonin for modulating the synapse is very interesting, but maybe that's a topic for another time. Okay, so GABA and glutamate, we think, are in the same vesicle. Now, some of you here are probably thinking, well, you know, Bernardo started this talk telling us about action selection, and now he's telling us about biophysics. And so why? What's the connection here? And so what I hope to do now is switch and show you this very preliminary data in which I want to convince you that, in fact, this has a lot to do with action selection. Okay, so remember, dopamine signals that something good has happened, or that something good is expected to happen, and changes the brain in such a way to repeat the action that led to that favorable outcome. And the system has to learn to do this, right? So plasticity in the brain is what allows dopamine to gain this reward prediction error encoding. Now, as I mentioned, habenula inhibits dopaminergic neurons in the ventral tegmental area. And so therefore, it has to learn the opposite. It has to learn to predict that something bad is going to happen and then alter the brain in such a way to avoid it. And so we believe that this synapse here between the endopeduncular nucleus and lateral habenula is a key site of plasticity that mediates that learning on rapid timescales. And the idea is basically this. We think of cortex as basically knowing everything, right? So it has access to all the sensory information. It knows the motor actions that the animal's carrying out. It knows the context. It knows internal drive, right? Whether the animal's hungry or thirsty or so forth. And it provides that information to striatum, which combines it in such a way that striatal neurons now represent some combination of these pieces of information coming from cortex. And that further gets refined based on the convergent structure of synapses across the basal ganglia in such a way that then the endopeduncular nucleus provides to the habenula a distilled set of pieces of information that's gained from cortex. And now the lateral habenula basically has to say, is this thing something that predicts good or bad outcomes? And we speculate that it does so by regulating this GABA versus glutamate transmission. So if we step back a bit, what's really fascinating about this synapse is that the sign and weight of this synapse is set solely by the numbers and kinds of postsynaptic receptors that are inserted. So the synapse releases GABA and glutamate, the presynaptic side, but the postsynaptic side can choose to insert GABA or glutamate receptors and make this an inhibitory or an excitatory synapse, exactly the way artificial neural networks work. So we can think about this in a different way. The endopeduncular nucleus provides information to the habenula. The habenula calculates a negative value, okay, negative because of the signs of the synapses here, which is fed into the ventral tegmental area, which then compares that to a true value. And the difference of the two is then the reward prediction error, the value minus the predicted. Now, if that, and that's, of course, encoded by dopamine. You know, other people have other proposals, but there's a lot of evidence that dopamine encodes RP, at least from the VTA. Now, if that information of this error signal is provided back to the habenula, then that is exactly the learning signal that one needs to teach and train the synapse by so-called gradient descent in order for the habenula to encode value perfectly. I'm not going to show you how this is done. It works. For those of you that care about the math, you can go and calculate and do backpropagation of differentials on your own. But the point being that this synapse between the endopeduncular nucleus and the lateral habenula, if it's exposed to this error signal, can then learn to calculate value based on this very simple rule, which is that one should update the synapse between the EP and the lateral habenula based on this three-factor learning rule, which basically says, if a synapse was active and a postsynaptic cell was active, when something unexpectedly good happened, then we should weaken that synapse. And if it was active when something unexpectedly bad happened, then we should strengthen that synapse. And of course, this makes sense because the habenula inhibits dopamine, so it should tend to bias something towards more activity in the habenula, less dopamine if something bad happens, and vice versa. But the point being that this really is a formal learning rule that will make the habenula learn the perception of value. So seen in a different way, we can think of all of the synapses between a somatostatin neuron of the EP and a habenula neuron as ranging from purely GABAergic to purely glutamatergic, and they're shaped by the following learning rule. If a synapse is active when something good happens, then increase the GABA receptors in that synapse. And we can measure that in vitro with whole cell physiology, as I showed you. And this should enhance dopamine release, which we should measure in vivo versus with D-light. On the other hand, if a synapse is active when something bad happens, then increase glutamate receptors, which again, we can measure in vitro, and this should suppress dopamine signaling, which we can measure in vivo. Okay, so how can we test that? Well, this is where I'm now going to transition to preliminary work that's been done by Shun. And so what Shun did is he placed a head-restrained mouse who was thirsty on a rig in which he could hear a cue, okay, or was delivered rather a cue, and session to session, we changed the pairing of that cue to predict a good outcome or a bad outcome. Okay, so we could induce rapid plasticity session by session for that cue outcome association. Okay, now the cue that we delivered in this case was a combination of a tone and optogenetic stimulation of endopeduncular habenula synapses. Okay, and the reason for pairing these together is that by putting the cue and the tone together, we had something salient, which we could pair with a good or bad outcome. And then we could cut an acute brain slice and activate those same synapses and ask, what's the synaptic transmission look like? Or alternatively, in vivo, we could activate those synapses now without the tone, without the reward, and ask, what effect does that have on dopamine release? And is that altered by experience? Okay, so does experience alter the ability of activating these inputs on releasing neurotransmitter in the habenula, exciting the habenula, inducing dopamine release in the VTA, and altering GABA glutamate transmission in the slide? I'm not going to walk through the full behavioral test, but it consists of a habituation session shown here on the left, random pairing between the cue and different outcomes so that the animal experiences all possible contexts and outcomes and cues without any information, and then switching to sessions in which we have positive pairing or negative pairing. And interleaved in all these, we have catch trials where we don't give an outcome, or trials in which we just deliver the optogenetic stimulation to probe what those synapses are doing and what state they're in. Okay, so I've mentioned Shun, and as I mentioned, small n, work in progress, take it with a grain of salt, but I think it looks really exciting. And remember that all of the data I'm going to show you is from delivering just the optogenetic stimulation, not the tone, just the optogenetic stimulation in these probe trials to see what happens to the effect of those synapses on the brain. Okay, so animals learn the task. So here you can see an animal that's trying to lick. Here comes the optogenetic stimulation, and then it only licks when the water reward comes out. It doesn't have any anticipation. Here you can see late trial, and again, the animal's waiting. Okay, and you see the optogenetic stimulation come on. It started licking immediately, and that's because it's already learned the association that water is available. Here on the bottom, you can see the air puff. Optogenetic stimulation comes on, and then it wipes afterwards. Here's the air puff, and you can see right there, it's now come to anticipate it, and it closes. Okay, so if we look at the several animals, we can understand that the animal is learning the task by just looking at the anticipatory licking that occurs with the optogenetic stimulation, okay? And what you can see is that as we switch an animal from positive pairing in blue to negative pairing in red, that it starts to suppress its anticipatory licks, and then when we switch back to positive pairing, the animal again resumes its anticipatory licking to the cue. So it's definitely changing the association of the cue with the presence or absence of water, and we can see that effect by changing anticipatory licking. Okay, so what's the analysis that we've done in the circuitry? So in this case, we're expressing channel rhodopsin in these endopeduncular somatostatin cells, and then we're analyzing what's going on in the habenula or in the nucleus accumbens. The first data I'm going to show you is analysis with a glutamate sensor of glutamate release in the habenula that's evoked by optogenetic activation of these EP inputs, okay? And what you can see is that as the animal learns to pair an optogenetic stimulation with a good outcome across a first session or a second session in groups of 10 trials, what you can see is that the glutamate release driven by that optogenetic activation is not changing. Okay, so release of glutamate doesn't seem to be changing at that synapse. And you can see it here by just piling them on top of each other. Across these two sessions in which we see behavioral changes, there's no change in glutamate release from the EP inputs of the habenula. Now we can move further downstream, though, and do exactly the same experiment, except now record dopamine release in the nucleus accumbens with D-light. And so what do we see there? Well, we see that before pairing, if we optogenetically activate these inputs, this evokes no dopamine release and no suppression of dopamine in the accumbens. This opportunistic stimulation really has no value for the animal, as one would expect. On the other hand, if in that same animal we give a water reward, we see that we evoke dopamine release in the accumbens, and if we give an air puff, we inhibit dopamine in the accumbens, just as we would expect for a positive and negative reinforcing experience. Okay, so now what happened now in the context of the behavior of pairing? So here you can see early pairing trials in which we have the optogenetic stimulation and then the water reward. And you can see that the optogenetic stimulation evokes no dopamine release in the accumbens, but of course, on the water reward does, okay? But as we continue to pair it, now the optogenetic stimulation itself gives this anticipatory dopamine release in the accumbens as if it's now gained a positive value. And of course, I showed you before that in parallel with this, the animal starts doing anticipatory licking, showing that it actually has attributed some kind of positive value to this stimulus. Now, what happens when we switch from the positive pairing session to the negative pairing session? Well, here's the first 10 trials right after doing that switch. And what you can see is that there's some residual positive going dopamine release in the accumbens to the optogenetic stimulation. Of course, this was previously paired with something good and now something bad. And then when the air puff comes on, we see inhibition of dopamine. As we continue this pairing through groups of more 10 sets of trials, you can see that now the optogenetic stimulation begins to inhibit dopamine release ahead of the air puff, and then the air puff does more. Again, the valence of this optogenetic stimulation, which is just activating these fibers, switches from evoking dopamine release to suppressing dopamine release with this pairing, just as we predicted, and as I'll remind you, without altering glutamate release. Okay, so here's what that data looks like. This is summary data in these groups of trials that are shown on the right. And what you can see is that before pairing, we activate these optogenetic expressing somatostatin inputs in the habenula. It has no effect on dopamine release. After pairing, it inhibits habenula. We haven't shown that yet, but this causes dopamine release. After negative pairing, it will excite habenula. Again, I haven't shown that yet, and it should inhibit dopamine release. Now, I proposed that the site of plasticity was this synapse here. And so do we have any evidence for that? So remember this three-factor learning rule. It says that if a synapse and a cell are active, when something unexpectedly good happens, we should weaken that synapse, make it more GABAergic. And if something unexpectedly bad happens, we should strengthen that synapse, make it more glutamatergic. So this is an example of a whole cell recording in the habenula at -70 millivolts to isolate the excitatory current, or at 0 millivolts to isolate inhibitory current in an acute brain slice, in which we optogenetically stimulate a large set of inputs. This is not single synapse analysis now, this is population analysis. And you can see the glutamatergic input and the GABAergic input. Now here's three such currents, three such cells from a naive animal that's had no pairing. You can see inward current and outward current. And if we take 20 or so of those recordings and look at a bootstrap distribution of a subset of those to understand what the natural variation is in the log of the iPSC to EPSC, you can see that distribution shown here in blue. And these dashed lines show the cutoffs for p equals 0.05 for things that are statistically different from the mean. So what about now for paired animals? Well, if we do exactly the same experiment after positive pairing, you can right away see that it looks like the glutamatergic current is diminished compared to the GABAergic current. And this is what we would have expected from the three-factor learning rule. If we take the cells that we recorded from this condition and plot them, you can see that the iPSC versus EPSC is greatly skewed compared to the naive population in blue. And if we show that in this bootstrapped histogram, you can see it lies far, far out on a tail, very different than one we expect by the mean. In fact, this is p 0.0001 or something like that. So clearly pairing has altered the nature of the synapse in vivo, consistent with what we predicted, which was driving it towards GABAergic and modulation of dopamine. So I'm sorry, I must have talked very fast because I've showed you a lot of stuff, but I'm at the end of my data. So where are we now? Well, as I mentioned, this is preliminary analysis. So we have to do a lot more of it, but we're very encouraged by what we're finding. We think we are showing evidence that GABA-glutamate transmission ratios change with positive pairing is what I showed you. We're going to do it now for negative pairing. We think this occurs without altering actual release of GABA-glutamate. This is a postsynaptic plasticity mechanism, but we have to really prove that, and we're going to work on that. In terms of the theory, we'd like to understand if there really is an optimal GABA glutamate distribution that might serve as a substrate for plasticity. And that's something we have to do a little bit of theory on. I've shown you now evidence that pairing opsogenic stimulation with something positive drives the synapse to be GABAergic. But I argued, though, that this really served to calculate value, which of course is an analog signal that can vary from something very good to a little good to very bad to a little bad. So we have to think about how we can actually get at that value encoding as opposed to simply positive versus negative valence encoding. And so that's something we want to look at. And then lastly, of course, we need to do some sufficiency experiments and understand if we disrupt these plasticity mechanisms, can we disrupt rapid updates in value estimation? Now, I want to just reinforce that what I'm talking about, I think, are these rapid updates due to contingency changing and not long-term estimations of value that happen with very repeated exposures, which of course we expect to happen more upstream, for example, at corticostriatal synapses. We think this might be the primary site of learning, which is then transferred further back with exposure. Okay, so as I mentioned, Solat Kim did the GABA glutamate co-release experiments. That paper was published last year and she's defended. Shun is carrying on now with the behavioral work that I've shown you now. And Wengang Wang is a very talented staff physiologist in the lab who does whole cell recordings and is supporting Shun in the analysis that we've shown so far. Mike Wallace started the analysis of EP when he was a postdoc in the lab. Sarah was a very talented electrophysiologist postdoc in the lab who's now in Austria, who helped Solat get started. We've had a variety of co-ops, technicians, and other people in the lab who supported this project, a variety of funding sources that I'm showing here on the right. I'm happy to take questions.
Excellent. That was, okay, super. That was really interesting. So let me just start with a couple of questions, Bernard. So first of all, the model for co-release, I think that implies that essentially the packaging is the same. There's no variation from vesicle to vesicle in the GABA glutamate ratio. Is that a fair statement?
Yeah, so that's a really interesting question, something that's very, very hard to get at. I think there's a couple of ways to think about it. So on the one hand, you'd think, well, if a vesicle has 5 glutamate transporters and 1 GABA transporter, maybe you're going to get more glutamate than GABA. I don't think that's the case. I think we need to think about this as an equilibrium state. So if the transporters run to equilibrium, then they should achieve the same GABA concentration vesicle to vesicle, independent of how many transporters they have. Whereas the kinetics, how quickly they reach that equilibrium should depend on transporters. So I think the variability in the currents probably has more to do with changes in synaptic vesicle size, which the radius changes plus minus 10%, which is like a 30% variance in volume. But it's going to be really hard to get at that. And if you have good ideas of how to do it, I really welcome it because we're struggling to figure that out.
Yeah, interesting. So that question, so you mentioned serotonin. So this gets at the older and sort of somewhat crude idea about serotonin and dopamine opponent effects, right? And I guess your comments seem to imply that this would be a potential site for that kind of interaction.
Yeah, so what I'm really interested in there, and I hope to get at, is that if serotonin weakens endopeduncular to venula synapses proportionally, independent of whether they release GABA or glutamate, then the way to think about that is that it's essentially saying, let's give less weight to our past experience, right? And let's not let what we learned in the past influence dopamine signaling as much. So I think that that's something we really can test in the same kind of paradigm that I showed you now, where if we give serotonergic agonists or delete these receptors or so forth, we should be able to see the effective experience weigh more or less on the actions of the animal. And so for that, we have a 2-arm banded task. That's a information integration task that we've done a lot of mathematical modeling. So we know how the animal uses past experience. And we would predict there that if we give serotonin or potentially SSRIs or some 5-HT1B agonists, we should be able to make the animal go into a state where it pays less attention to its past experience and simply uses present cues to make a decision. Well, we'll see.
Okay, interesting. Yeah. So past experience over what time interval? I mean, how long would that be? So
yeah, good question. Don't know. I don't know.
Okay.
I think what we're seeing in this contingency switching task, it looks like sort of 10 kind of experiences is enough to do something here. But yeah, we'll see.
Okay, interesting. Very cool. Open for other questions for Bernardo, since we have a little extra time today.
Yeah, sorry if I went too fast
No,
there.
no. That was really interesting. I really enjoyed that. Was really enjoyable. So I think, well, in that case, I think we'll give people back a little extra time today for their lives. But again, I want to thank Bernardo for taking the time to give a really, really interesting talk. And
Thanks, Roger. My pleasure.
yeah, look forward to seeing this in print when your student is a little further along. So
A little further along, yeah.
okay, thank
you.
you.
Take care. Bye-bye.